So guys, so if you guys has ever like done a wet lab, so I believe you guys has ever felt like experienced this. Like the first one, you guys need to do a repetitive work, like it keeps failing and then it's very laborious. And then the next one, you guys will find a lot of human error, and then it fails, and then you need to redo it again, and it might takes like weeks or maybe months, or even like in worst case maybe years of doing the research. And which is very tiring, especially because like most of these stuffs are from basic research, which should be easy but takes a lot of time. That is why uh, we're joining this hackathon, because like using Biofoundry is very uh useful for this. And there is this particular topic that we want to explore, which is antibiotic resistance, because especially in Indonesia, despite the Minister of Health ever, so a lot of people are using antibiotic because it's very practical. Like you just drink the antibiotic, and then you're healthy, and all the bacteria is dead. Like your infection is gone. So people keep using it, and then it becomes irrational, and then they will get antibiotic resistance, and then we need to find another natural resources to make the antibiotic. Like, to be honest, Indonesia has a lot of biodiversity, and that is why a lot of people are researching on this. Like Indonesia is big, like, and almost every universities are doing this. So like there are really a lot of people and money coming around like on this topic. So we want to propose about using the biofoundry, because like to do the natural product research, we only need to use several basic research, which is of course very laborious. So by changing it into biofoundry, especially the frugal one, because biofoundry is expensive, we want to use this to assist our antimicrobial drug discovery from Indonesian natural resources. So to do this, there are several steps that we are using. The first one, we want to collect the plant materials, of course, by human. And then the second one, we will collect the compound by extracting the samples that we have using our automated metabolite extraction system. And then the second one, we will screen all the compounds that we have. And then we will do antimicrobial susceptibility tests and to find the antimicrobial drug candidate. And from all these two information that we have, compound collection and antimicrobial drug candidate, we will store it into our database. So to be honest, even though database is very important, very useful, and a lot of countries have been using it, somehow Indonesia doesn't have it yet. So we want to make it. And then from the drug candidate, maybe like others, maybe startups or institutions or like big company, pharmaceutical company, they can use our base research to further develop to make their own product for the next clinical trial maybe. So we'll have a lot of innovation from this, and it might like make people flourish and be richer, I guess. And then we want to dwell more to the our modules. So the first one, the extraction, and then the second one is hit identification, basically to find the drug that we want. So the first one, to do it, so we want to first we need to store our samples. So of course using the refrigerator, and then the next one we need to green the samples, and then we do extraction using solvents. So this is the approach that we want first, because this is very easy. And then the third one is to heat it in hot plate, and then finally we want to centrifuge it. And after we get the compost, we will store it in the freezer again. And then for the heat identification, it's very easy. Basically, we test our extract to the bacterial sample that we have cultured before, incubate it, and then we check the OD to check the concentration, whether there's a growth or there's no growth. So like from all the equipments that we are using, so because we want it to be cheap, because like Biofoundry is very expensive, so we either use secondhand or by using do-it-yourself. So because like equipments like freezer and blender, we can find it like almost everywhere in every home. We can find it. That's why like, and it's very functional. It's very good. We can use it in the lab. So it's better to opt the secondhand than making it ourselves. And then second one, the DIY, like spectrophotometer, and then centrifuge, and then hot plate. Basically, we find some open source DIY protocol that wants to help us. And actually, especially for enzyme, you guys like have a presentation that tells us like how to make spectrophotometer, which is very helpful. Thank you. thank you for that. And then for some equipments like incubator and then the automatic pipetting system, like it's actually I've found like how the DIY to do it. And maybe we can like do some stuff using Arduino or stuff like to make them, but I think it's very difficult. So like maybe we can opt for a second hand, or maybe if there's donation, maybe it'll be very helpful. So like the importance of our biofoundry here is because like a lot of researchers in Indonesia like revolve around this natural product, and a lot of like commercialization or startups or companies, like even food companies, are using this, especially beauty products, because like there are a lot of them and they have a lot of money, so they can do a lot of researches. And I believe like a lot of them really will need like help from automation to do their research, which will be very helpful for them. So I guess this will be impactful for Indonesia and maybe the world, I guess. Thank you very much.
Awesome.
That
Great to
was
hear from
really
you.
awesome. Yes.
All
Thanks
right.
for
Any
liking the
questions?
comic.
Yeah.
Yes, thank you for the great speech and staying on time. That was a great 5 minutes, almost 5 minutes. Now we're
nice.
opening up for questions, so go ahead. And I can ask the first question if no one's going to do that. How are you going to make this biofoundry idea tailored to specifically antibiotics? And is there any specificities that you need for antibiotics aside from other things?
So here it is. Like, actually, if you look at our modules, so we make it as modules. So if we want to change like our approach in our research, so we can just change the module and like stack it with another equipment, and we can do another research using it. So the first one is our extraction module and hit identification. So we separate both of them. So in case if like there is a company or maybe institution who wants just to do the extraction, we can use like our first module only. And then what's your other question? I'm sorry.
Oh, it's just making it specific to antibiotics instead of a general biofoundry. If there's any specific hardware that you need aside from this.
So actually, we're just using the spectrum. So all the equipment that we're using are actually, to be honest, is very general. Like spectrophotometer is, you can use it for everything, but like that is enough to do the MIC method first. So to be honest, like why do we use this first is because it's very simple. So we want to find something that's feasible first. Then once we can do it, then we will go further and doing like the more complicated stuff.
Any other questions?
I had a question. Yeah. So this is a problem that you told is very prevalent in Indonesia, right? So there have been a lot of people working on this. So have you identified what problems they run into, and is this biofoundry implementation of this problem solve any of the current problems that the other guys are facing?
The problem that we want to tackle mostly revolve around like that the work is very laborious and it takes a lot of time. Like if we use Biofoundry, I found out that like, if I'm not mistaken, is it Gingo? So they can do like 100,000 of results like a day. Well, if you're doing it ourselves, it might take weeks, I guess. So if you like make it faster, we can make more drugs for that, or maybe other products. So that's like the main reason we want to do this for the laborious work.
Sorry, may I help?
Oh yes, please.
Yeah, basically, if there's still time, yeah. I mean, I just want to add that the idea is because Biofoundry can do all of these things in automated fashion. We use robotics, right? And then we have a lot of research already about natural products for drug discovery in Indonesia, but all of them are done in the lab, wet lab by people. And then, as we mentioned before, there's no database that curated all of those results. So we hope that by having a Biofoundry, we can, let's say, redo all the research, for lack of a better word, but now we're going to have a better data quality because all of them will be precise, will be reproducible. So it will be, it may lead to a better drug development later on. So that's why we think Biofoundry is really crucial to make sure that all the knowledge and all the resources that we already have by now, it can be actually translated into an actual product. Because as for now, we're still importing a lot of drug material from other countries. So we need to also think about that, how to make Indonesia able to supply its own drug materials and especially antimicrobials.
All right.
Sounds great. So a follow-up question on that, not a follow-up, probably a little bit independent. So do you, if you have to implement a biofoundry like this, do you guys have the expertise among the team that you have right now, or do you know people who will be able to make this happen? Or
You mean in our current team right now?
yes.
Oh yeah, we have Farrell here, one of our member that is doing internship in drug discovery, and also we have Eri. Also, he's a pharmacist, so he also has an expertise in drug discovery, including antimicrobials.
So Sir Isan has also gotten like decades of research experience
I
as
also
well
have
for
some
this.
basics in microbiology, so maybe I can help in some direction. But
Okay.
for like implementation, we will need expertise from outside, obviously.
All right. Awesome hearing from you guys. And we'll be getting in touch through Joggle, and we'll see the descriptions, and other teams will be peer reviewing this. But awesome to hear from you. We've gone to 5 minutes of questions.